Okay, hello everybody. Welcome back to our second session here at CCNS. I'm going to turn things over to Itai, who is going to be running this session, and he'll introduce our speaker.
Hi everyone. So this session would be about neurons and microcircuits. We have a few speakers and one tutorial, and it should be very interesting. We'll start with the keynote speaker because it's late in Israel, so we don't want Miki to talk at midnight. So a few words about Dr. Miki London. He really nicely combines computational and experimental neuroscience. He used to work with Idan Segev, then Michael Hauser, and then started his own lab back at the Hebrew University at the Edmond and Lili Safra Brain Center. He's an associate professor. And a lot of very interesting things, representing single neurons as deep learning networks, and studying computations in the dendrites in relation to processing. So it's my absolute pleasure to welcome Miki to present. The stage is yours.
All right. Thank you, Itai. And thanks the organizers. It's a new thing for me. I've never did such a lecture on Crowdcast, but I hope it will go smoothly. And I want to talk about two projects today. I won't get into too many details in both of them because I want to cover both, but the idea is to talk about one computational and one a little bit more experimental project. So the title of the talk is What Makes a Neuron Spike? It's a very strange question because we all know the answer, right? And the outline will be we're going to talk a little bit about this question just to set up the stage for what we do. And I will present you then a work by a student that I co-supervise with Idan Segev, David Ben-Guyev. And then we'll talk a little bit about dendritic computation, and I'll present here work on dendritic neuromodulation of two of my students, 2 or 3 students, Yair and Adi. And then we'll make a short detour about neuromodulation in general, which is a work of Shiran, and we'll conclude maybe if we'll have time with another aspect of this question, which is a work of a PhD student of myself, Amir. So Yair has just completed his PhD, and David and Amir are completing it now in these days. So imagine we just kind of consider a neuron. This is a nice picture of layer 2-3 pyramidal neurons from a human cortex that we recently did some patch clamp recording from. Imagine that you could, of course, in human we cannot, but in rat or mice we can record the membrane potential. And we look at this membrane potential, and from time to time the membrane potential shows a spike, an action potential. And we ask ourselves, what really happened that led to this spike? And this is a very fundamental question in neuroscience, maybe the most fundamental one. And of course, many people worked on that. And we basically know the answer, right? The answer is that there is a threshold for action potential. When the membrane potential crosses the threshold, there is a spike. And what makes this spike, or what makes the membrane potential cross the threshold, is basically that there is some inhibition and excitation coming to the dendrite. And whenever there is enough excitation, large, then the membrane potential depolarize enough to cross the threshold, right? So what makes a neuron spike is simply the activity of the network. There are massive activity of other neuron, which enforces the membrane potential to cross the threshold and then makes the neuron to spike. And if you ask most theoretical neuroscientists, especially the physicists, they would just tell you that the network activity is so overwhelming that it basically washing over all the individual properties of the single neuron. In some cases, they will also talk about the fact that this excitation and inhibition kind of cancel each other, and there's some balance. And then the deviation from this balance are the things that making the membrane potential cross the threshold. But this is just a nuance. It's an important fact, but it's a nuance on the same idea that you have more excitation than inhibition. And if you look for evidence for this network dominance view, you would find many of them. First of all, that neurons are usually considered to communicate via firing rate. And everywhere where you look for function of neuron, if you stick an electrode and present some stimulus or look for motor activity, you would see that changes in firing rate, which works well with this idea of changes in massive activity of the network. The responses of neuron to the same stimuli, even if you control very well the stimulus, it's highly variable, which also supports this view of balance of excitation and inhibition. And there are also some experimental works recently from the fly navigation system, like the ring model, or some experiments of Karl Svoboda and Charles Buchmann that shows attractor neural networks in the mouse brain. And lastly, I want to mention also things that was mentioned in previous talks, that deep networks that basically use rate coding, if you really kind of want to make some analogy to the brain, are very powerful and can actually show very good functional systems. So all this evidence support the view that neurons communicate via rate coding, and what makes a neuron spike is actually some temporal imbalance between excitation and inhibition. However, even if you look at this recording that I just showed you, and you'll zoom up to the first spike, you will notice something quite strange, which is that the membrane potential actually went above what seems to be the voltage threshold, but it didn't spike. And then it did spike at some other time where the membrane potential was lower, smaller than that voltage. So this already kind of shows us, and actually even in this one trace that was recorded from rat in vivo, anesthetized rat in vivo, already in this picture you would see more than once such examples, which kind of suggests that maybe that's not the whole story. And I want to highlight how difficult it is to address this question from a technical point of view, because if you really wanted to know what made the neuron spike, you would need to know, first of all, what are the inputs to this neuron, and this probably means you have to know about 10,000 presynaptic neurons, which are there, and where do they connect to the dendrite, and are they excitatory or inhibitory. You will have to monitor all of their activity to know each one of the spike, when did it happen, and did it actually activated the synapse, because we know that not all synapses are activated every time a spike is coming. You will need to know the synaptic efficacy of each one of these connections. You need to know the dendritic properties, which we know, but only to some approximation. And you also need then to have some theory to combine all this information, a giant amount of information, to know how all this input was converted into this spike, and why some configuration make spikes and some other do not. And this is really, really a difficult question. It looks as if we already kind of passed that question, but we are not even close. Just to make kind of clear where we are, I want to show you one of a recent paper of Eder Ranz that used to be in a PhD together with me in Mike House's lab. And he did this beautiful experiment where he patched a neuron and transfected this neuron with a rabies virus, allowing him to see all the presynaptic neurons to all the presynaptic neurons that are synapsing on this pyramidal neuron in the visual cortex, and it could actually map the visual responses of these neurons to drifting gratings. But even then, after this really difficult experiment, when you patch a neuron and then you kind of can see all the presynaptic neurons, you find out that, first of all, you can only trace about 150 to 650 neurons, which are about 6.5% of the input to this neuron. And you cannot actually record, certainly not the voltage from each one of them, and you don't know what they synapses onto the neuron. And some of these neurons are actually located in the thalamus, so you can't even record from them or image them or know much about them. So even with these very sophisticated techniques, we're still far away from answering this question. And there are other directions how to answer this question, but all of them amount to a lot of missing information. So this is a very difficult question. Okay, but we do know from some experiments in slices, from actually many experiments in slices, that the answer is going to be quite complicated, because dendrites have all kinds of voltage-dependent channels and other type of ion channels that makes a summation of synaptic input more complicated than we think. So, for example, in this work from Marty Larcombe, he showed that synaptic input to the dendrite can generate calcium spike in the dendrite, which then generate bursts of action potential in the soma. And Jackie Schiller showed that if you activate on layer 2-3 pyramidal neurons and also on layer 5 pyramidal neurons, if you activate close by inputs, you can generate an NMDA spike in the dendrites. And other people have shown that in CA1 pyramidal cell, for example, you can actually have forward propagating spikes if you have some depolarization in the way that allows the spike to propagate. So a dendritic spike can propagate to the soma and generate a spike. So if you were just to record in vivo this voltage in the soma, the membrane potential in the soma, and see a spike, you could just think that there was more excitation than inhibition, but in effect what happened was that there was some local spike in the dendrite that was propagated into the soma, and this is what made this spike. So there could be many other mechanisms that are going on in the dendrite that contributes to the generation of a spike. And then the answer to what make a neuron spike could be as trivial as depolarization, or if you ask an undergrad, probably the answer you'll get is optogenetics. It could be the network, but it could also be coincidence of time excitatory inputs, or coincidence of excitatory input that leads to NMDA spikes, and it could be coincidence of NMDA spikes that leads to calcium spike, and it could be some controlled inhibition or disinhibition. And recently also the evidence that there is like plateau potential that can act as switches that actually can put the neuron in a regime where it will respond to input or not respond to input, completely dependent on the selection process. So you could actually bombard the neuron with the same amount of input or exactly the same input. And once it will spike, and in other conditions it will not. So the answer to this question seems to be much more complicated than we wanted it to be. Okay, so there is one way, and we are, after all, in a computational neuroscience conference. There is one way to try to answer this question, and this is using models. For example, compartmental models. We can take the neuron, reconstruct it, have a model of the neuron, incorporate many ion channels and currents in the dendrite, and then we can also add a synapse that receives some spike train, and then we can predict what would be the output to this activity of the synaptic input. And I like to think about it as a Turing test for a neuron, because after the work of many people, and recently a lot of this work was done by Itai, we have a very good model of neurons. And in a sense, it's such a good model that if I'll show you this output trace, spiking and membrane potential and spiking trace of this neuron as response to the activity of this input, you would not know if this is actually an experiment that was done, or a recording that was done, or a simulation, because the simulation can actually fit every experimental piece of data that we have from these neurons. So we feel that the compartmental model have passed the Turing test for a single neuron in some ways. Okay, I have to remind you that that we haven't done the full experiment, so we don't really know what happens when you activate all the synapses or many of the synapses, because we've never done them experimentally, so we cannot test the model against this data. But from all other points of view, it looks like it's a good model. And then it at least allows us to pretend that we can overcome some of the technical problems that I just mentioned earlier. Okay, so what would it look like? It would look like something like that. So this is a neuron, and we bombard it with many synaptic input, excitatory and inhibitory, and you see that the membrane potential from time to time have action potentials, the dendrite have calcium spikes, and in general there is like a sea of activity in the dendrites, many small NMDA-like spikes in many dendrites, and big waves of activity that sometimes create action potential in the soma and sometimes do not. But the key point is that the activity is very complex. The output only shows a little bit of this complexity. And what we really want to understand is how is the input that bombarding this neuron are causing this output of the neuron. And the compartmental model gives us a very useful tool to ask this question, but eventually we are left with many differential equations that we just have to run to see the output, and it doesn't give us a lot of understanding of what is the process that happening behind the scenes. So recently we had this project that used deep neural networks to try to answer some of these questions. And the idea is that for the first time, we suddenly have a tool that can actually fit pretty well almost, or not almost, but many complex input-output functions. So if we have an input to this neuron and we have the output, maybe the deep neural network can actually find the mapping, and then at least we'll have an alternative to the compartmental model. And maybe it will be useful and maybe not, but it was trying, see if it actually can work. Okay, so the idea is that we take a compartmental model. It actually is a model of Itai. We generate many spike trains of excitatory that activate excitatory synapses and inhibitory synapses. We sample them from some distribution. We play a lot with a relative balance between them. So you see there's a period of high excitatory and medium inhibitory input, and then quiet, and then some other activities. So we play with all of them, and then we get for each simulation like that, we have an action, membrane potential and action potentials, membrane potential and action potential trace that we record in the soma, and then we can ask a neural network to map, to try to find the mapping between these spike trains and this output. And what we want is the network to predict at any millisecond the membrane potential and whether there is a spike or not in the neuron at a millisecond resolution. And this is a giant task. We have kind of made hundreds or thousands of simulations like that, and generated a very big database. And then we try to fit it with different neural networks, and eventually with a temporal convolutional neural net with 7 hidden layers, we managed to get to a pretty good fit. Okay, we set some criteria. I'm not going to go into the details, but you see that the network in cyan can actually, in magenta, can predict almost perfectly the membrane potential and subthreshold membrane potential of the neuron, and also when it spikes. Okay, so this is a very good fit, and we were kind of happy about that. And if you just want some kind of evaluation, we can kind of make an ROC curve. We can do a cross correlation between the input and the output, and you can see also the membrane potential of the model versus the predicted membrane potential of the network, and you see that it does a very good job on all of these parameters. Moreover, this network actually generalized very well. So if we kind of activating only parts of the dendrite, so we target the neuron, we take now a simulation and we put the excitatory inputs and inhibitory inputs only on some of the dendrites, not on all of the dendrites. And we run the simulation and then we run the network. They still kind of fit very well, even though they haven't seen any such example in the training set. Or if we introduce some synchronization in the input, excitatory, inhibitory input, still we have pretty good, we see like bigger deviations, but we still have a very good fit between them and in other conditions as well. So the model generalized very well out of the distribution that it was trained on. So now we can actually look at some of the input units to this network. So if we look at the first layer of the hidden units in the network, we can see interesting things there. So we can kind of read out of this network of these filters that the network is using. We can see some of the things that the network learned. For example, it learned to separate the excitatory and the inhibitory inputs from each other. Even though this information was not given to the network, it kind of realized that some spikes are inhibiting the neuron and some spikes are exciting the neuron. And even you can see like features that shows you that these passive properties of this to this neuron. So for example, some inputs have high amplitude or contribution and time constant, and some of them are slower and smaller, probably relating to distal synapses, very proximal synapses. And this is the basal inputs. These are the oblique inputs. And you see that this unit specifically is completely insensitive to the apical tuft. And here's another unit like that kind of cares about excitatory and inhibitory inputs coming to the basal dendrites and to the oblique dendrites, but less to the apical dendrites. But there are also some other units that care about the apical dendrites. And here in the apical dendrites, we see that they care less about the temporal dynamics. They don't care exactly when the input comes, but they do care about if it comes somewhere in some wider window, temporal window. Interestingly enough, we can also ask why do we need so many layers? So in this case, we have several layers, so we can try to remove some of the features of the compartmental model and see what contributes to this complexity. And we were kind of shocked to see that if we remove the NMDA conductances from the dendrites and we replace them with AMPA-like conductances, so in general, the total conductance is more or less the same, but we don't have the nonlinearity of the NMDA currents, then a network with even one hidden layer is enough to get very good in terms of like the same amount of accuracy as the original model, as a 7 layers to the NMDA model. Okay, so the NMDA synapses are key to show this complexity of the dendritic tree. And the reason is quite clear, in the sense that the neuron actually, the network predicts the membrane potential very well, but what you really care about is what happens around the threshold. And you want to minimize the variance around this area, because this is where you're going to miss. If you're going to miss, you're going to make a mistake in predicting the spike. And when you have an NMDA, a neuron with NMDA current, then you have to make the prediction of the voltage precise, not only in the soma, but you have to make it at any given branch, because each one of these branch, if you miss, you can actually miss an NMDA spike, and this NMDA spike has a big influence on the chance of having a spike in the soma. So the network needs to work much harder in order to predict the membrane potential and the spikes, because it needs to know the spike at every given, at every branch at any given time. And it cannot make any mistakes there. And this is a very difficult task to do. Okay, so one question you can ask me is what's special about the number 7? And in order to address that, we made many simulations with many networks, sizes and depths, and number of neurons. And what you can see is that if you have AMPA and NMDA, you have a cluster of networks that actually requires about 7 or 8 layers. And sometimes you can even do good job with 5 layers, but in general you need many more layers than if you have only the AMPA synapses. And of course, if you have AMPA synapses and you do use 7 layers, then you get very good fit as well. So in conclusion for this part, I just want to show you that the deep neural network can actually learn to replicate the input-output transformation of realistic neuron at the millisecond temporal resolution. The network generalizes very well to unfamiliar input patterns. The filters that the network uses can actually tell us something about what the neuron does. And in some ways, it does some kind of a template matching to the synaptic input. And the activity of NMDA synapses significantly increases the complexity of the input-output transformation. So there are many other questions that we can ask. I will kind of skip that for the benefit of the time. But I want to tell you that you can go to Kaggle and look for the page of David. His username is Selfish Gene, so you can find him easily. And all the codes and the simulation is there and very, very organized, so you can play with it yourself and study this, and maybe can tell us that you can do it with fewer layers or better architecture, and we will be happy to be challenged on that. And lastly, I want to show you one thing that we are playing with now, which is very interesting. And the idea is that if a neuron is equivalent to a deep temporal convolutional network, we know that these networks are actually pretty powerful. So, for example, can we use that to see if a neuron do something useful? For example, distinguish pictures of cats and dogs. So the idea is that you take this neural network and you teach it to distinguish cats and dogs, which is pretty obvious, but it's not that obvious, because in our case, we don't want to change the weights of these hidden layers, because these hidden layers represent the transformation that the neuron does. So all the dendritic nonlinearities and stuff like that. So we don't want to to get into that. So we only allow to change the first layers, the synapses between the input and the first layer. Okay, but then this becomes a more difficult task, but it's doable. And if you do that, ideally, if you manage to do that, then you can actually map these inputs to the neuron, and then you can present the neuron with a picture of a dog that it hasn't seen in the training set, and ask the neuron to tell you whether this is a cat or a dog. Okay, so this is we thought that this will be pretty straightforward. It's not that easy. We're still working on that, but we hope that we'll manage to get that working, and then we'll have an example of a non-trivial task generated by or made only by a neuron, a single neuron, using its dendritic nonlinearities. Something that I think we don't have a good example of that today. Okay, so now I want to switch to a different story. It's related, but it's different. It has to do with neuromodulation in dendrites. So as I told you, the pyramidal neurons have this calcium spike in the dendrite. This is one of the most prominent features of these neurons, and we are going to focus on this calcium spike. And this has been shown by Matthew Larcum in 1999, where when you have interaction between inputs to the dendrite and inputs to the soma, you can generate a calcium spike in the dendrite that depolarize the membrane potential in the soma and generate a burst in the soma. And what is interesting about this phenomena is that these inputs that arrive to layer 1 is special kind of input. And not everybody appreciates that, but one example for that was a paper by Abbs et al. At 2018 that showed it also used the rabies virus tracing method, but from interneurons that resides in layer 1. There are not many of them. There are 4 types of these interneurons, but if you target one type of them and you map over all the brain where the inputs coming to these neurons, where the axons that coming to these neurons come from, you will find that many of them are coming from every place you can think of in the cortex. So from sensory cortex, auditory and somatosensory and visual cortex, but also from the prefrontal cortex and the orbitofrontal cortex and other places in the cortex, but also from the thalamus, and also from some midbrain and midbrain areas. So the input that arrives to this layer 1 is very rich, and it's very different from the input that the thalamic input that comes to layer 4, for example, or to the other layers of the cortex. And this makes this coincidence detection between the somatic input and the dendritic input a very interesting case, because basically you can start thinking of these neurons as doing some coincidence detection between local input, like feed-forward input, that arrives to the somatic and oblique area of the neuron, and then context input that comes from the rest of the brain to the apical dendrites. And Matthew Larcum wrote a very nice review about that, showing that, for example, you can think of the ways that the brain integrates information is constantly pumping in input to the lower layers, feed-forward input to the lower layers that combines from higher visual areas, for example, in this case, that process motion that project back to V1 to the apical dendrites. Okay, but you can also combine here some sounds and other features like color, and then basically perception is a constant ping pong between feedback, like prediction, what you think is going on, and what you actually see, and you combine that, and then this propagates up to higher areas all the way. So this is a very nice story, but this story is kind of presenting a very static picture of what is going on, because in some ways we know that when we process information, sometimes we are very aware of the context, but sometimes we are concentrating on what's coming from the visual input and we don't pay attention to the context. Sometimes we actually ignore the visual input and only kind of dream or daydream and think and process like internal information. So what we were looking for is some mechanisms that can actually change the coupling between these compartments. And one idea came through neuromodulation. So I will show you some examples, experimental evidence that shows that norepinephrine, and recently also we think of acetylcholine as something that can play a role in how much the neuron pay attention to each one of these compartments or to the coupling. So just to remind you that norepinephrine comes from the locus coeruleus. It's a brainstem nucleus, and it projects everywhere in the cortex. And if you look in the cortex very to axons of norepinephrine, you see this very dense mesh of these axons. And if you put it together with the neurons, you see that they are kind of, the dendrites and also the somas are never too far away from an axon of norepinephrine. If you zoom in, you will see pictures like that. So you see these axons running around the dendrites, and sometimes you even get a picture that looks as if the axon wrap around the dendrite. We don't know if this is a real thing or just an accident, but it's certainly interesting. So one thing that we did about that was to do calcium imaging of the dendrites in an awake mouse. So the mouse is walking on a treadmill, and we do calcium imaging from GCAMP from layer 5 pyramidal cells in barrel cortex, and we see the dendrites, we can trace them, and then we can pick up region of interest at some plane of focus around the branching points where we know that calcium spikes happen, and then we look at what is the calcium signals in this region of interest, and we see calcium activity. It's not very common, but from time to time you see these big calcium events in the dendrites. And then we add a drug which is called guanfacine, which is an alpha-2A adrenoreceptor agonist. And we do that on purpose, and I'll tell you in a second why, but this kind of imitates increasing the concentration of noradrenaline in the cortex. And when you do that, you pretty quickly see there's a big elevation in the frequency and amplitude of these calcium events in the dendrites. And moreover, we can even spread the drug from a pipette and monitor what happens, and you see that the dendrite lights up when we do that. So we kind of conclude that activating noradrenaline increasing the excitability of these dendrites. And building on previous knowledge of experiment by Wang et al. From 2007, there is the idea that these alpha-2A norepinephrine receptors, they are localized very close to HCN channels that we know that have high density in the apical dendrites of these pyramidal neurons. So the idea is that when you activate this alpha-2A adrenoreceptor, it blocks cyclic AMP, which is required for the opening of HCN channels, and then the channels are blocked. And we have less IH in the dendrite than compared to the case where we don't have noradrenaline. So what happens if you block IH, similar to what the noradrenaline is supposed to do? Then you also see increase in the activity of the dendrite. They become more excitable. Okay? So we see that this guanfacine increases the excitability, When blocking IH increases excitability, and the question is, does guanfacine actually block IH? And to do that, we did some patch clamp in vivo in these neurons. And when we do that and we apply guanfacine, we see that the typical sag that is recorded in the membrane potential, which is a signature of IH, disappears when we apply guanfacine, which is an indication that indeed guanfacine blocks IH, but it doesn't actually block it, because if you hyperpolarize the membrane potential enough, then you still see the sun. So it's more consistent with like shifting the activation curve of IH to the left. So if you are at some membrane potential, you kind of close IH, but if you go enough hyperpolarization, then you will see this IH again. So we can model that, and we can see that if we block IH, then we have hyperpolarization, and then the chance of having a calcium spike is bigger, and the calcium spike will be bigger. And this is actually very similar to results that have been shown by Mark Arnett, when he worked with Jeff McGee and Stephen Williams. But then in a simulation, we can actually test what happens if we kind of shift the activation curve to many values, and then when we do that, we see that we have a gradual change in the excitability of the apical tuft. So basically the idea is that when you apply norepinephrine to the dendrites, you can actually change the excitability of the dendrite and control how sensitive is a neuron to this activity that coming from the dendrite. So I'm going to skip, because we are late, I'm going to skip a few slides that I wanted to show you on recent projects that we did to image the activity of noradrenaline in the dendrites, and I'll just skip to the last bit, which is kind of surprising. And what we do here is we do imaging of in green of the axons that carry noradrenaline that comes from the locus coeruleus, and in red we image the dendrites in the barrel cortex. And strangely enough, we activate and we stimulate the mouse with an auditory signal. Okay, and what we see is that, you'll see it in this movie, that when the sound comes, the axons are activated, and then a little bit later the dendrites are also activated. Okay, so we get like both the noradrenaline signal and the dendritic excitability. And then when we look at them together, we see that the norepinephrine is responding to the sound almost every time, but not every time with the same amplitude. But the dendrites are either responding to the sound, which is kind of weird to begin with, because these are dendrites in the barrel cortex and they respond to sound, but sometimes they don't respond at all. Okay, sometimes they're active with no correlation to the input. So sometimes the sound triggers the dendrite and sometimes it doesn't. So what we plot here is that what is the probability of the dendrites in barrel cortex to respond to sound as a function of the activity of the noradrenergic fibers. And what you see is that the larger the noradrenergic fiber is, the higher the probability of the dendrites to respond. So in a way, our interpretation, of course it's not the only interpretation, but our interpretation is that the noradrenaline, when it comes, it makes the dendrites more excitable, and then the auditory input that arrives through these layer 1 axons can trigger the dendrites, and then you see activity in the dendrites to sound. And moreover, if we inhibit the locus coeruleus, then this probability drops. So if we kind of apply this HM4DI, which when we inject the mouse with CNO, and this inhibits the locus coeruleus activity, then the probability of the same dendrites to respond to sound decreases significantly. Okay, so I just want to conclude now and kind of wrap it up with some demonstration. And the idea is that we have the context input coming to the apical dendrite and the recurrent and feed-forward input coming to the proximal region, but they are disconnected from each other because of this IH activity. But when you have norepinephrine, this norepinephrine closes the IH or modulates the IH and makes it less active. And this allows the dendrites to be more excitable. And then you have this ping pong between the context and the feed-forward input. And this is can be, of course, be different depending on the level of norepinephrine activity. And if you want to put it in this picture of Matthew Larcombe's Savannah story, so imagine that you go in the savannah and you see a picture, you look around, okay, and you have some visual input, but you don't see anything special there. But then you hear a roar, and this roar actually goes to some auditory areas and then to prefrontal areas, and you realize that there is a lion around. Okay, but the roar is also making something else. It of course make you very alert, and it activates an noradrenergic system, and then you have noradrenaline around, and you have this feedback input that tells you there's a lion around, and this modify the processing of this neuron, and it kind of helps you identify this lion because you know that there is a lion around. So once you know there is a lion, it's easier to spot it. So I want to just conclude everything with saying that there are strong evidence for unreliable neurons modulated by massive network activity, but there's also significant evidence for sophisticated and combinatorial synaptic integration that relies on dendritic nonlinear mechanism. We don't know the answer yet, and we won't know it for some time, but maybe some technologies like voltage imaging in vivo that is starting to show maybe will help us see the truth. And I want to thank my lab members in recent hikes that we made. And Yair did all this noradrenergic work together with Adi, and David did this deep neural network study. And Amir is working on acetylcholine. And Stav and Ben are working on mouse ultrasonic vocalization, which is a completely different story that I've haven't talked about. Thank you. And if you have questions, I'll be happy to answer.
Thank you, Miki. That was really fantastic. It's a very nice illustration of both the computational and experimental work that you're doing. We have a few questions. I can read them. Let's start with a question from Francis Skinner. Why do you think the apical dendrites, unlike the basal and oblique, do not separate excitatory and inhibitory inputs, if I followed correctly? Could it be because the biophysical channels distribution in the model on the more extensive apical dendrites need to be modified?
I'm not sure I'm following. So the question is, does in the deep neural network, the network kind of thinks that maybe I didn't present it well, but it's not like. So if the question was about these filters that has like inhibitory and excitatory inputs, it's not that these are coming to the basal and oblique. So these three columns are the filter of one input unit of this deep neural network. And this is time. So the network actually is sensitive to input that happened recently, more than input that happened, like spikes that arrived way before, like 80 milliseconds before. and it also sensitive to excitatory and inhibitory inputs that arrive to the basal and oblique dendrites, and this specific unit is less sensitive to inputs that happens in the apical dendrite. And if we zoom into these units, I don't have the picture here, but if we zoom into that, it looks as if it does something like that. So it really cares about the strong input that arrives right before the spike, and then less and less to inputs that arrives a little bit before that. And in this case, this tells me that most of or many of the spikes can be predicted by what happened in the recent time window of the basal and oblique dendrite. So many spikes can be predicted just by looking at this basal dendrite. But some spikes, you need the contribution of what happens in the apical dendrite, and in that case, they less care about the proximal basal and oblique dendrites. And why do the network kind of do this separation? It's a good question that we don't have the answer. So I want to say one general thing about that, which is maybe a wishful thinking, but I'm still very wishful about it. Many of you know that about 10, 15 years ago, almost all computer viruses came to Windows computers and never to Linux and Macs. And the reason was that there weren't many Macs around and there were many Windows. So people bothered to write the viruses to Macs. So my hope is that because so many people are doing neural networks now, then we'll have tools to understand what they do, and there will be more effort in doing that than compared to the effort that people are doing to understand neurons. So if we have a good representation with deep neural network, maybe we'll have the tools to analyze it that we'll never have in neuroscience. So maybe that's a wishful thinking, but in deep neural networks, sometimes when you look at these kernels, you don't really understand what they're doing. But at some point, we will hopefully will have these tools.
That's great.
So I don't know why the network is doing the separation. I really don't.
Yeah, good answer. So another question from me, actually. So you showed that when you remove NMDA, it's enough to have one hidden layer, right? The question is, did you try what happened if you remove calcium channels? How many layers are sufficient to represent the neuron?
No, it's a good question. We should do that. Yeah. We haven't done that. What we did do, but I don't remember the answer, but I can look for it. We did put the inputs like here. Sorry. We did do the case where we limit the inputs only to the basal dendrite, then to the basal and oblique, then to the whole neuron. And then asked how many layers do we need to do that, and I don't remember the answer, but it's in the supplementary figure. So then you still have the calcium channels, but we don't have any inputs coming there. So probably there are no calcium spikes. So we can look at that, and I can send you the answer, but I don't remember.
Yeah, cool. All right, good. I'll look at the supplementary. Good. A question from Gabriel Benigno. Thanks for the great talk. Have the detailed multi-compartmental models been assembled into a network? If so, how does the network activity differs from simpler LIF model?
So the first of all, the answer is Itai has done that a few years ago. There's a very nice paper about that, and also this is a part of the Blue Brain project. So the Blue Brain has all these neurons with many other neurons, but it's worth looking at these papers, and I think they do have some comparisons between networks of full compartmental models and LIF neurons. I don't remember the big differences, but I think they do point that there are some differences. in the dynamics of these networks. For us, what is interesting is if indeed these neurons are seven-layer deep neural networks, what will happen when you start making networks of these networks, and how would they actually behave? Because it's a kind of a special architecture that with some constraints that people do not consider so much. So we are working on that as well in context of learning and plasticity and dynamics, but we don't have good answers yet.
Okay. A question, I think, probably from Scott. How much efficiency is added by using this deep neural network rather than simply simulating the multi-compartmental model?
So it's difficult to compare because one is using GPUs and the other don't, but we have a factor about 2,000 faster simulating the neuron using a deep neural network. So you can actually speed up like the the blue brain simulation 2,000 faster is really, really significant if we could do that. But before that, we really want to know that we are good enough in order, because otherwise you'll kind of generalize faster, but with wrong answers. So it's a good question, but I think we can have a big speed up using these models.
That's great.
Totally.
Another question potentially from Scott. If IH activity impairs dendritic excitability and in turn integration, how might that explain these findings that misexpression of IH contributes to various forms of epilepsy? Is it related to IH contribution to excitability, or perhaps more complicated relationships related to dendritic properties?
Yeah, so there is some evidence, especially in the hippocampus, that mutations to IH can contribute to epilepsy. There's a researcher called Mala Shah, which is working on that in University College London. If I'm not mistaken, she's very famous for that. We have a feeling that that the interpretation that we have is that IH contribute to the probability of having an dendritic action potential, and then acetylcholine, there's a very recent interesting work by Stephen Williams, can contribute to the duration of this excitability once it was started. But the relationship is not obvious, so we don't really know what the key players. It could be, for example, the low threshold activated calcium currents that are actually mediating this effect, because when you block IH, what you do is you remove a lot of or release a lot of low threshold calcium, voltage-dependent calcium channels from inactivation, so they are they are available, and then any depolarization recruits them and helps the calcium the calcium spike to start. So it could be like a more complicated chain of actions. So IH affect low threshold calcium channels, and these channels are contributing to the probability of spike being initiated. So this is still an open question, and the problem is that we know very little about the distribution of ion channels in the dendrites. No one bothered to actually do this work. It's very difficult work. So there are a lot of unknowns, and the models are great, but they don't give us the full resolution that we need to understand what's going on there.
Yeah. A question from Alex Guetmcwright from my lab about the, because IH, when you block it, it will hyperpolarize the cell, right?
Yeah.
So he said, it seems odd that when you block IH, it leads to enhanced excitability, while at the same time it does hyperpolarize the cells. It sounds like this could possibly be explained by enhanced calcium spiking, maybe. But can you also expand a bit on the effects of HCN block in inhibitory neurons, maybe, and the effect on the network?
Well, this is, of course, everything here that I've presented, and I should have said that more clearly. We don't have a direct evidence that the norepinephrine is working on the dendrites of these pyramidal neurons, because we can't do that. So it could be that norepinephrine or IH is modulating inhibitory neurons, and these are the ones that are modulating the pyramidal cells. And we are working on that, but we still don't have good evidence for that. But this is an experiment that was done in pyramidal cell in slices, and they patch the dendrites, and you can clearly see that when you apply ZDs, there is a hyperpolarization of the membrane potential, and this is actually very shocking hyperpolarization, because if you calculate the amount of channels that are supposed to be open at minus 55 millivolts, this is where IH is already kind of almost closed. So if these IH channels or HCN channels that are open at minus 55, which is about 0.5% of the total amount of channels, you block them and you get 15 millivolts or 20 millivolts hyperpolarization, it means that there are tons of these channels in the dendrites. And then you see this hyperpolarization, but still when you depolarize the membrane, you get bigger spikes compared to these calcium spikes. And this replicates pretty well in the model. And we suspect that what happens here is that when you hyperpolarize, you, as I said before, you release low threshold calcium channels from inactivation, and then these kind of contributes to the upstroke of the spike, and they recruit more calcium, high voltage calcium channels. So then you get a bigger spike. But we don't have a good understanding yet of this process, as I said. But I think at least in the slice it doesn't involve inhibitory neurons. But it replicates nicely in the model also without any inhibitory neuron. So it's probably an internal property of the pyramidal cells.
Very nice, very interesting. So there are a couple more questions, but I think we'll stop here. Mickey, thank you so much. It's been very illuminating, and looking forward to chat with you. Yes. And we will be in touch about a lot of those things. So Scott, I'll let you continue.
Yeah, so we're going to have a very brief break here. So Itai will be the next talk at about 2:05. So Itai is going to go set up, and we'll go live in that session in a couple of minutes. So we'll see you all there.
Great. Thank you.